Welcome everyone, and thank you for joining us for our presentation titled Towards Antibody-Based Treatment of COVID-19, a panel of human neutralizing monoclonal antibodies targeting SARS-CoV-2 spike at multiple epitopes. It's my pleasure to introduce to you Dr. Mazor, who is the head of the Department of Infectious Diseases at the Weizmann Institute, and he earned his doctorate from the Weizmann Institute for Science at Rehovot, Israel, where he conducted his thesis study on developing photodynamic-based drugs for cancer therapy. For complete biography of our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we want to encourage you to submit them to the Q&A box, and our speaker will address those questions following his presentation. And as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. Please join me now in welcoming our presenter, Dr. Ohad Mazor. I will now turn the presentation over to him. Welcome, sir.
Hi. So today I will tell you about our efforts to develop antibody-based treatment for COVID-19. First of all, just let me tell you who we are. So IIBR is a governmental cutting-edge research institute that specialized in the fields of biology, chemistry, and environmental sciences. And we have a national and international collaborations. In the biology division, we focus on studies of different bacteria, toxins, and viruses. So let's go into antibodies. So we wish to let the evolution do its best to make the best antibodies, and the way that the evolution do it is by affinity maturation in vivo. So it starts with a VDGA gene recombination, and then upon exposure to an antigen, there is a somatic hypermutation, mostly in hotspots across the CDR's region, the parts of the antibody that binds the antigen, and the resulting antibodies have improved affinity, and upon a second or third exposure to the same antigen, these whole cycles go on and on until a very high affinity antibodies are reached, mainly in the reaching a ceiling affinity of the nanomolar range. So in our group, we isolate high affinity antibodies that enable specific neutralization and sensitive detection of target antigens, mostly toxins, viruses, and bacteria. And our methodology is to obtain peripheral circulatory lymphocytes from human patients or from immunized animals. And then we isolate high affinity antibodies using phage display libraries. We generate full-length antibodies and characterize them. Just a quick overview of our methodology. So this is the antibody. We are talking about the viable region that is expressed as a single chain. Both the VH and the VL are expressed as a single chain antibody on the surface of a filamentous phage, the M13 phage. We create a library that contains hundreds of million different phages or different antibodies, and then by a process termed panning, we select only these antibodies that specifically bind the antigen. This process can be repeated several times in order to enrich and specifically isolate the desired antibodies. The way to develop or to create such a good library is to amplify the genes that encodes to the whole variety of antibodies from the donor both to the heavy chain and variable region and to the light chain variable region. We are talking about both the lambda and the kappa. In order to do this, we have a specific and very large repertoire of primers for each species. We then assemble them randomly together to create two libraries, one for the heavy chain with the lambda chains and the second for the kappa chains. And then we sequence many clones of each library in order to ensure that it is diverse. We're using the Sanger sequencing sex studio system. And just for example, after a second round of planning, we test random clones from the library. And as you can see, there are several clones in each library that bind to the antigen. And after the third round, most of the clones usually bind the antigen, some of them with a very high affinity. We then reformat the antibodies to a full-length IgG. So this is an antibody. So we are using for all our antibodies a constant region, a human FC IgG1 type region, to which we join the variable regions that were derived from the phage display library at the end of the planning process. We then express these antibodies in CHO or in HEC293 cells, purifying them, and further characterize them. Basically, in our laboratory, we are focusing on pathogens that are biothreat associated. But in the last few months, we have isolated a panel of human antibodies that we hope will serve as the basis for combined post-exposure therapy for SARS-CoV-2 infection. So SARS-CoV-2, this is the virus. You are all well familiar with this. We are specifically want to target the spikes. And what we aim to do is to isolate antibodies that target different epitopes on the spike protein. So we obtained lymphocytes from recovered COVID-19 patients. We also obtained from immunized animals, but today I will focus on our human antibodies. We created the library, and now we are characterizing the antibodies. So we have penned the library using the receptor binding domain. This is the moiety of the spikes that specifically bind the human ACE2 receptor. And we cloned we sequenced the resulted clones, reformatted them into IgG. And by the end of this project, we ended up with 8 different antibodies that belong to different germ lines, both for the heavy and for the light chains. These antibodies specifically bind to the receptor binding domain, which is part of the S1 region of the spike. As you can see, it does not bind, they are not binding the N-terminal domain, for example. We then wanted to measure their affinity. So we are using Octet. It's a biolayer interferometry system. The antibody in question is immobilized to the sensor. And then we add the antigen, in this case, RBD, at different concentration. And we measure both the K on and the K off part. And then we can calculate for each antibody the affinity, the KD. And as can be seen here, all our antibodies have a quite good affinity, which is in the low and even sub-nanomolar range. We also asked whether our antibodies can bind the antigen simultaneously, or in other words, whether they bind to different epitopes. So we again are using the octet system. So the first antibody in question, for example, MD65, is immobilized on the sensor. And then we add the antigen, in this case, the S1. And as you can see, upon binding of the S1, there is a very nice shift of indicating for the binding to the antibody. And then we watch the excess. And then we add another antibody. So if the second antibody binds to different epitope on the antigen, then it will result in another wavelength shift as shown here. If it binds to the same epitope, then we will have no further shift. So we characterized all the antibodies, and we could classify them into 4 groups. 2 of the groups only are represented only by 1 antibody, and 2 groups have 3 antibodies. And basically, what we are seeing here is that the small moiety, the receptor binding domain, contains at least 4 different epitopes. We then asked whether those antibodies can neutralize the virus. We are using in vitro system to test the neutralization on Vero cells. And as can be seen here, basically 7 out of the 8 antibodies can neutralize the virus. We have also added here a control antibody, the RB18, an isotype control. And just want you to focus on our best neutralizing antibody, MD65, which belongs to the second group. And also, you can see that the antibody 62 from group 3 is also have a very high neutralization potency. We then asked whether we can say something about the mechanism of the neutralization of these antibodies, again, using the octet. So the first antibody is immobilized to the sensor, and then we add the receptor binding domain, and then we add the receptor, recombinant receptor, human ACE2, and we test whether this receptor can bind to the RBD in the presence of the neutralizing antibody. So we took a representative antibody from each group. And as can clearly be seen here, is that, for example, antibody 29 belongs to group 1, does not inhibit the binding of the receptor to the RBD, and so does antibody 47, while 2 of our best neutralizing antibodies, antibody 65 and antibody 62, completely abolished the ability of the receptor to bind to the RBD. So this can tell us about the most important, maybe most important part of the neutralization mechanism needed for SARS-CoV-2. However, please bear in mind that antibodies 47 and 29 both inhibit the activity of the virus in vitro. So I guess maybe probably 2 other important mechanisms. So where we are now, so we are continuing to characterize the activity of the antibodies in vivo in animal models. We also perform preclinical evaluations, both pharmacokinetics, toxicity, etc. We are currently also characterize another set of human antibodies and antibodies that have been derived from SARS-CoV-2 immunized animals. And with that, I would like to thank the people in my group and all the other people who helped us to the antibody engineering group, and to our colleagues from the virology unit. And thank you all for listening.
Thank you, Dr. Mazor, for that outstanding presentation. And we will now move to the live Q&A portion of the presentation. And as a reminder, please submit your questions via the Q&A box. So Dr. Mazor, we have already some existing questions coming in. Our first question from our audience member is, have you tested your antibody panel against any of the SARS or MERS viruses? And if so, what type of inhibition did you observe?
Hi, Susie. Thank you. So unfortunately, we did not test our antibodies against the other SARS or MERS viruses. If there will be cross-neutralization, I guess it will be with the SARS, which shares high homology with the SARS-CoV-2 virus, but I don't know about the MERS. I guess for the MERS, it probably will not work as well.
Thank you so much. And I want to thank our audience members for coming today and remind them that any questions not answered today will be answered via email. Dr. Mazor, based on your data, do you think that when this gets used in clinical trials, you would need to do like a combination of antibodies? How would you determine which set of antibodies to include?
Well, that's a very good question. Basically, once we have several antibodies against several epitopes, we can combine them. Whether this will be the best way to start, I don't know. I guess we will wish to start with one antibody, preferably the best one, I guess MD65. And then we'll see what happens. But if we have to combine them, I guess we will take the most neutralizing antibody, which is MD65 and MD62, which can also bind simultaneously to the virus, so they can act together, whether synergistically or just as a combination. But I guess this will be my two best candidates for a combination therapy.
Thank you so much. And we have one more question coming in live. Can you talk a little bit more about the next steps in which could be a normal or expected timeline in the preclinical and clinical steps?
Yeah, so basically the preclinical steps will include, first of all, toxicity studies and pharmacokinetics in different animals. And what we are doing also right now is to try our antibodies in vivo. We have a mouse model, the transgenic human ACE2 mice model, that we are currently trying to see whether our antibodies can provide therapy for COVID-19 infection of these animals. So this is where we are now, and we hope to establish the preclinical steps in a few months and go to the clinic.
Thank you so much. And Dr. Mazor, thank you for your time today, for your presentation, and also for your important research. And I want to thank our audience members for their outstanding questions. Dr. Mazor will be moving into the expert lounge right after this talk to continue discussing his presentation and answering questions submitted during the presentation. And this presentation will be available on demand viewing through 12 months from today. So all the way up to next year, 2021 in October. And please share it with your colleague who may be interested in today's topic. Thank you again for your participation, and see you in that expert lounge. Take care, everyone. Bye-bye, and stay healthy. Thank you, Dr. Mazor.
Thank you. Bye-bye.